The last class, what was taken on the basis of the structure of DNA, its calculations are important. I think you have understood it, and which was very important on the basis of CBSE examinations or any other examination that is related to biology. So now we are leaving this concept, and now we are entering into another concept which is called as central dogma. The word dogma means it feels or it thinks it is correct. That means that is the only way. That's the meaning. That's the only way to carry something from one place to another. That's the meaning of dogma. And what is this important of central dogma in genetics is whatever the DNA is there, finally it has to convert it into a protein, because DNA is the language that should be converted into a non-message, and with that non-message the cell has to function accordingly. So this DNA is protected or preserved inside a code, and this code has to be opened up and it should be converted into a non-language. The code is DNA, and the language which the cell knows, especially the ribosomes knows, is on the basis of mRNAs and tRNAs, and the ribosomes will convert it into a protein. So that protein will carry the information or the command what a cell has to function. So the first thing itself, if you see there are three things are given: DNA to RNA, RNA to protein, and there is a red line is given to the top, that is RNA to DNA, which is known as a reverse transcription. And you can see there a circle around DNA, a semicircle around DNA, which is said that DNA can replicate by itself, and RNA also, it says that RNA also can replicate by itself, but proteins can't replicate by itself. Proteins has to be synthesized according to the language of DNA. And why it is protected? Because many other organisms are there in this world which can read our DNA, and they can incorporate into that particular host DNA, and they can override the system of us, and they can run. So one of the best example, which is called as the AIDS virus. AIDS virus is a retrovirus. What this HIV virus is doing is, HIV virus will come and infect to one of the human cells, and it will transmit their DNA, transmit their genetic material, which is an RNA. This RNA is having, that means that the virus is having an enzyme known as reverse transcriptase enzyme, which can synthesize a complementary DNA according to the RNA, and that DNA have the capacity to incorporate with the human cell, and that it will replicate inside the human cell. Instead of producing a protein that is useful to us, they will produce a protein which can produce which will produce the young ones of HIV virus, and this HIV virus will go and infect to other cells. That process will continue. That is why HIV virus is considered as deadly. Once we get it, we will be most of them will surrender to death, or otherwise if we are staying in a good condition environment, then our life expectancy will increase. So first before entering into what is DNA or DNA to RNA, RNA to protein, the translation and transcription process, we will discuss one thing: DNA. We have already studied about DNA, and we will study here about how DNA can replicate by itself, and is there is any experiments that has proved that DNA will replicate by this method, like that. So we will study that. Then the next class we will study about RNA, then protein, that is how DNA is converted into RNA, how this RNA is converted into protein, what are the changes taking place, those things we will study later. So today we will study about the DNA replication. So scientists believe that after 1952 and 1953, when the first time the structure of DNA was predicted by Watson and Crick. And this central dogma, this concept is also given by Watson. Francis Crick published this central dogma, and they also believed that DNA can be replicated. That means DNA has to replicate, because when one cell will divide it into another cell through mitosis or through meiosis, the division of chromosome is very important according to the mode of cell division. If it is a mitotic division, it will be equal number of chromosome or equal number of genetic material, and if it is a meiotic division, there will be only half number of genetic material, or half chromosomes will be present. So it is very important for the division of genetic material. So genetic materials are inside the chromosomes. So scientists propose 3 theories by which replication can be taken place. One they call it as semi-conservative, conservative, and dispersive model. These are the 3 models that was proposed. The first model was proposed as a conservative model. So if you see in this conservative model, the parental DNA is there. This parental DNA have the capacity to generate 2 different type of DNA. That means according to the parental strand, they can synthesize 2 different type of DNA, and that can be through the basis of heredity, it will be expressed in the generations to come. Then next comes the dispersive model. The dispersive model, if you see that, it is 50-50. That means some part of the parental DNA, that means 2 parental strands are there, that will be mixed less randomly, will be present inside the daughter cells. That is what that means few codons or few ATGCs or few codons from the parental strand will be present inside the daughter cells. And next comes the semi-conservative model. Semi-conservative model states that one of the DNA, that means there are 2 DNA strands are there which are running anti-parallel to each other. Both the DNAs, one among the DNA strand will be conserved inside the daughter cell, and according to that a new strand will be, adjoining strand will be grouped, or they will be attached together and it will form. That is why if you see this in semi-conservative model, the parental strand is having a pink color strand, but when you see this first generation, you can see 50-50, and when you see in the second generation, you see that there is a violet color strand is also there, a mix of parental strand is also there. That means the grandparental strand is also there. So these are the 3 replicative process what was proposed by the scientists. Among this, accepted model of DNA replication is semi-conservative model through the basis of an experiment did by Michelson and Stahl. So Michelson and Stahl conducted one experiment, and this experiment was to find out whether the DNA is replicating semi-conservative, conservative or disruptive method. So what they did is, instead of using some traditional methods, here they have used radioisotopes according to their heaviness. That means they have used N14 and N15 hydrogenous bases, because nitrogenous bases are present inside the DNA. So that particular nitrogenous bases are replaced in the second generation by either by N14 or N15. So this was their understanding, and after the bacteria started to use this N14 and N15, they will do a density gradient centrifugation. That means by using cesium chloride, they will go for the density gradient centrifugation. So this was their idea. So what is their understanding is, if one strand of the DNA is N14, so whenever they are centrifuging, that means they are rotating this in a very high RPM, so different layers they will get inside a test tube. That means on the N15 and N14 model. So here what they did is, this is what they got. So one of the test tube, all the DNAs were initially labeled with N14, then they are allowed it to grow inside the N15, and the samples were taken. samples were taken and they have done the ultracentrifugation. So in ultracentrifugation, what they found is in the 0th minute or in the 0th time, that means in the there was no growth was there, 100% of the strands were of N14. And when the first generation, that means in every 20 minutes E. coli will replicate, the first generation came at first 20 minutes when it was they found it was 100% of N15 and a little bit of N14. But when in the second generation, that is in the 40th minute, when they found, it become 50-50. And in 60th minute, the parental strand, that is of N15, they started to reduce, and the hybrid strand N15, they started to increase N14, they started to increase its number. So from this they have concluded that in all the generation, that first generation, second generation, third generation, fourth generation, fifth generation, a small percentage of parental strand was seen in each generation. So from this they have concluded their studies that DNA replicates by a model, by a process known as semi-conservative model. That means one among the parental strand will be preserved in each and every generation. So this is what they are finding. On this basis, now everybody understood that the DNA replication is going on the basis of semi-conservative model, which is done by Michelson and Stoll. Am I clear? So before going through what the diagram that is given in the textbook, better you go through this diagram which I have taken from the net. It is little more understandable than the book. Book is right, there is no mistake in what is given in the book, but book will make you little confusion because diagrams are not that much colorful, but this is little bit colorful, so it is very easy to understand. That's it. So this is a method. So now the question comes, how the DNA will replicate by itself. And here that explanation of that particular each and every step was given, what they have result they have got. So whatever explained, the same thing according to the Michelson and Stoll. So that in detail they have given. And next question comes is, how the DNAs are replicating. What all are the requirements for DNA replication? Because why this DNA replication become very important, because nowadays we can replicate DNA inside the laboratories. So if we want to replicate, if we want to synthesize a new DNA from a single strand of DNA, we have to know how naturally this synthesis is taking place. If we don't know that, it is not possible to create that exact step inside the laboratory, or it is not possible to make any type of changes inside the laboratory. That is one of the main thing. Is it clear? So what all are the requirements for the DNA replication or DNA new synthesis of strand? First thing is, we need substrates. Substrates means the basic things. For example, when you are constructing a house or a school, we need some basic stuffs. Like if you want to build a building, that is cements are required, particles of rock, bricks, different types of bricks in different numbers. So all like that, when DNA replication is also taking place, we need those type of things. The first thing we need is substrates. Substrates means we need ATP, GTP, adenosine, adenine, thymine, guanine, nitrogenous bases. Those things are required. If they are not there, a new strand is not possible to form. So the 4-deoxyribonucleotide triphosphates are required for the for this, that is DATP, DGTP, DTTP, DCTP. That means deoxyadenosine triphosphate, deoxyguanine triphosphate, deoxythymine triphosphate, deoxycytosine triphosphate. These are required. And why they are coming in between triphosphate? ATP, GTP. Why they are coming in the group of phosphate? Because when they are joining, they need energy. So when this phosphate group is cut, it liberates energy. With that energy, they can attach or into a new and form into a new strand. That is the main thing from that. Then we need a template strand. That means one among the strands of DNA. Then we need a primer. And after that we need enzymes. Because why enzymes? Because enzymes are biological catalyst. They can enhance the rate of reaction. So what are the major enzymes that is required for the synthesis of DNA? One enzyme is helicases. These enzymes have the capacity to open the DNA strand. That means they have the capacity to break the hydrogen bond between the base pairs. If the hydrogen bond is not broken, the enzymes can't read what are things inside. Then there is a single stranded binding protein. So we will discuss about this, what are the functions of this. Then topoisomerase, primases, then DNA polymerase 2, 3, and your textbook is given RNA dependent DNA polymerase. That is the term that is given in NCERT, RNA dependent DNA polymerase. So if you are writing RNA dependent DNA polymerase, that is also correct. If you are writing DNA polymerase 3, that is also correct. Or if you are writing DNA polymerase, if you are writing that, both are correct. There is nothing to worry about that. Then primases are there, then ligase enzymes are there. So helicases will cut open. Then DNA polymerase have the capacity to synthesize a new strand. And DNA ligase is the enzyme which joins the strand which is newly formed. Is it clear? Then other enzymes I will explain when we are explaining the process of this synthesis. Then there are different types of DNA polymerase are there. This is just for your understanding. CBSE is not going to ask any questions from the types of DNA polymerase. Because DNA polymerase family, there are 6 families are there, and in each family there are different types of polymerase. That is DNA polymerase 1, DNA polymerase 2, DNA polymerase 2, DNA polymerase 3, so DNA polymerase 4, lambda. So these are there. This is just for your understanding. And if you are somebody those who are writing for this entrance examination, there they can ask which is a DNA polymerase. They have both the capacity to replicate and repair. So DNA polymerase 1 and DNA polymerase 2, then which is have only the capacity to do only replication, that is DNA polymerase 3. So these few questions they can ask in your examinations. If from last 2-3 years this question is not asked, but there is a chance of asking this. That's all. So this is the complete process of DNA replication. So it is a hand-drawn picture so that we can understand it very fast. So DNA strand 1 is before that yellow and blue line. Remember, DNA strand is running in 3' to 5' direction. If one strand is in 3' to 5' direction, another will be anti-parallel to each other, that is in 5' to 3' direction. And every time whenever a DNA replication is taking place, these helicases enzymes, these 2 strands of DNAs are there, in between the hydrogen bonds are present. They will come and cut the hydrogen bond from the middle. When they are middle, these DNA strands will cut open. When it is cut open, then when this strand is cut open, that time a new protein will come and bind, which is known as SSB protein, that is single stranded binding protein. Why this protein is coming and binding there? Because when one strand is opened up, there is a chance that these strands may join together after some time, maybe or within a minute. So they want to avoid that, because now they have to do the replication. They don't want to join those strands together till the process of replication is over. So single stranded binding proteins will come and bind there first, so it will stop the joining of the parental strands. Then DNA polymerase enzymes will come and bind there. So before the DNA polymerase enzymes come and bind there, you must understand one thing. DNA polymerase will synthesize strands in 5' to 3' direction. So you can see that one of the strands is synthesized in 5' to 3' direction. New strand is synthesized. When that strand is synthesized in one direction, to the opposite direction also it can synthesize. So the strand which is synthesized in the 5' to 3' direction, that particular strand is known as the leading strand. And on the strand which is running on 3' to 5' direction, there also they will synthesize a DNA fragment, but those strands are not joined together. That means after synthesizing a particular amount of strands, there will be a gap, then they will start synthesizing. That particular strand is known as the lagging strand. So these lagging strands are also known as Okazaki fragments. And these lagging strands are joined together by DNA ligase. So this is the main thing you must understand. There is only one thing you have to understand: from which direction to which direction the DNA synthesis takes place. That is 5' to 3' direction. If the DNA synthesis has to take place, that means DNA replication has to take place in between 5' to 3' direction. So the parental strand should be 3' to 5' direction. That's all. So when one synthesis is taking place in another one strand, alternative anti-parallel strand also the same synthesis will take place. But on that strand, the 5' to 3' synthesis will take place, but that 5' to 3' synthesis is not in a continuous manner. There will be a gap will be there. So that particular strand is known as lagging strand, and that lagging strand is called as the Okazaki fragments, because the scientist who first said about this, that means when the DNA replication takes place, one among the strand in which the replication is running, there the strands, newly synthesized strand is formed in a discontinuous fashion. So they call it as the Okazaki fragments. And these Okazaki fragments will be joined together. Once it is joined together, before joining there is a proofreading. These strands will be checked properly. And DNA polymerase 2, DNA polymerase 1, DNA polymerase 4, they are very good efficient synthesizer. That means they synthesize new strand also. They check the newly synthesized strand. The error is 1 in 1 lakh base pairs. That is how they have said, 1 in 1 lakh base pairs. When 1 lakh base pairs are synthesized, then inside that one error will be there. So that is how neatly, strictly they are following the replication process. So this is what is called as the DNA replication. So if there is any doubt, please ask me. And this thing, it should be taught on board. I told that my difficulty. So please read this data, so whatever I have given it as a note, go through that. Any doubt, please read it. Or we are in the situation that the schools will reopen very soon. So that time I will clear the doubts, whatever the doubts are there. Otherwise, if you are understood the things, then you can ask me through online. What is the method? What are they? How they are formed, like that. If there is any doubt, you can ask me. and from Monday to Friday I will be sending MCQs, few questions of MCQs. That is also important because this time there is a more chance for knowledge based questions because of this corona. There's a chance, so we have to practice both theoretical, theory subjective, and the elective, both we have to practice. So any doubt, please let me know. So next week onwards we will start with the process of protein synthesis, how proteins are synthesized from one strand, from the DNA strand. Thank you.